Control of babesiosis. In case of Babesia bovis, it can be achieved by vaccination using live attenuated vaccines to prevent the occurrence of babesiosis outbreaks. To treat infected animals, drugs with babesicides are used like imidocarb, which eliminate the parasite within erythrocytes. An alternative to control bovine babesiosis is to control the tick vector population. Acaricides are used with limitations of acaricide resistance and environmental pollution. Another strategy is Pasteur rotation. There were also tick vaccines based on tick midgut antigen like BM86, which reduced tick viability and progeny, but it was removed from market. However, there are several anti-tick vaccines under development nowadays. Our lab focus is to develop transmission blocking vaccines to prevent parasite transmission. Let's take a look to the life cycle of Babesia parasites. Here we represent on the left an infected larva. When feeding on a bovine host, it will inoculate the salivary gland content, which include the Babesia sporozoites. Babesia sporozoites invade erythrocytes and transform into hemoglobin-feeding trophozoites that asexually divide into typically two pear-shaped merozoites and eventually lyse their host cell and invade new ones. Repeating this asexual propagation cycle causes hemolysis and anemia, which cause the clinical signs after 10 to 12 days, which called incubation period. When an adult female tick feeds on infected cattle, it will ingest some infected red blood cells. Variation in temperature, oxygen tension, or other factors present in the midgut of the tick help induce gametocide development. Gametocytes become extracellular and give rise to spiky ray bodies that form multinucleated aggregates. From these aggregates, elongated, emerge, and paired parasites fuse to eventually give rise to zygotes. Zygotes divide into kinetes and penetrate the tick midgut into the hemolymph to invade other tissues in the tick, such as the ovary, which cause the transovarian transmission. An infected larvae parasites will find its way to the salivary gland of the larvae, and when the larvae feeds, we restart the cycle. Sexual reproduction in the tick vector is essential for the transmission of Babesia parasites, but little is known about the behavior of gametes due to difficulties to obtaining purified parasites from tick midguts or lack of protocols to induce and culture these stages free of tick antigens. However, Babesia bovis sexual stages are newly characterized in vitro culture using an induction medium. The current study focuses on signaling events of sexual stage parasites during fertilization in the tick midgut. In order to describe sexual stages development and signaling events of Babesia sexual stage during fertilization, in vitro induction system for Babesia bovis sexual stages was used. Short-term Babesia culture recovered from acutely infected animal were used to induce gametocytes in vitro. When the parasitemia reached around 5% in culture, Babesia-infected erythrocytes were suspended in an induction medium containing xanthorenic acid and incubated at 26 degrees Celsius. The method is simple and yields high number of clean sexual stages free of tick antigen. Induction of the parasites was evaluated through Giemsa-stained microscopic evaluation. In figure 1 on the left, induction resulted in two distinct cell types, a large spherical shape and a smaller shape that displayed one or more projections. To confirm shapes observed by Giemsa scanning electron micrograph of Babesia bovis sexual stages in figure 2, showing similarity and exflagellation fusion, confirming the stages stained with Giemsa. Movement of Babesia between erythrocytes takes place by gliding, which is the typical motility style of epicomplexans, as you see on the left. Flagellates and processes are the movement style of gametes, which flagellae may help gametes find each other in the thick gut lumen in order to increase the chance of parasite mating, but their influence is limited to their length. It is insufficient at the long distances involved in the low gametocyte density blood meals. Alternatively, chemotaxis is a common long-distance solution to the problem of mate location in externally fertilizing organisms.